Thank you for such a kind introduction, and it's a great honor, and I must thank the Society for recognizing our work with this prize. And it's very flattering because I know that very great scientists have received it before me, and not less flattering as that I know the great scientists who failed to receive it, actually. Right, so we all know that antibiotics, besides playing an essential role in our everyday life, they're very powerful tools for enzymology, as investigating the modes of action of antibiotics can lead to some important insights into molecular mechanisms that are targeted by these antibiotics. And one of the antibiotics played a critical role in the quest for understanding how RNA polymerase active center works, the one that I'm going to present here. Right, I will remind you about the details of transcription. Transcription in all living organisms is accomplished by multi-subunit RNA polymerases, which are hugely conserved in all living organisms. And this is just an example of the crystal structures of archaeal, eukaryotic, and bacterial polymerases. And even though in archaea and eukaryotes some extra subunits have evolved after divergence of archaeal eukaryotic lineage and bacteria, the catalytic core that is made of 5 subunits is conserved through all domains of life. And this catalytic core, which accomplishes the synthesis of RNA, is made of 5 subunits, and in bacterial nomenclature it will be beta and beta prime, which make the pinces of the claw, 2 alpha subunits, which keep them together, and small omega subunits. So as I say, the 5 subunits make a structure resembling the crab claw, and within this claw, the nucleic acids are kept, and the reactions, the synthesis of RNA takes place. Right, so even though initiation and termination of transcription has diverged after the divergence of bacteria and archaeal eukaryotic lineages, the elongation of RNA and synthesis of RNA is almost identical in all domains of life. Elongation of RNA consists of repetition of so-called nucleotide addition cycle, which I represented here schematically. So the black is template DNA, the red is growing RNA, and the catalysis was for a long time thought to be solely performed by 2 metal ion mechanism. One metal ion is bound tightly in the active center of RNA polymerase by absolutely conserved triad of aspartates. The second magnesium ion is brought by the substrate. In this case, it is nucleotide triphosphate. So NTP binds in the active center and base pairs with the available template base. The catalysis takes place. The RNA is now 1 nucleotide longer. The pyrophosphate leaves, and RNA polymerase shifts forward to bring another next template base to the active center and vacate the space for the next nucleotide to come and to be incorporated. And this is the crystal structure representation of what I just said, and it looks quite similar to what I draw. Right, and RNA polymerase is a potent target for antibiotics, and we know more than 10 antibiotics that target transcription at the moment, and the most famous one is rifampicin. Obviously, you know about it, and which is clinically used. But for several of the antibiotics, the mode of action is not known, and during my postdoc in Konstantin Severinov's lab in Rutgers University in New Jersey, we were interested to elucidate the mode of action of antibiotic called streptolidogene. So we knew that streptolidogene, this purple thing here, binds quite far away from the active center. And this is the incoming nucleotide, the RNA, the template, and the catalytic magnesiums. However, if we do the reaction in vitro and we add the transcription in vitro and we add streptolidogene, we observe that nucleotide addition cycle was incredibly slowed down. So this constant shows you roughly how many nucleotides RNA polymerase can incorporate per second. So when you add streptolidinogen to the reaction, the nucleotide addition cycle slows down 4 orders of magnitude. This constant shows you the affinity of the nucleotide to the RNA polymerase, and it is not changed. When we saw these numbers, we suddenly realized that 2 years ago we had something similar, and it was actually my first project in Severinov's lab when I was doing part of my PhD in Rutgers. At that time, just a couple of years before that, we had the crystal structure, first crystal structure of RNA polymerase published. And it is a huge half megadalton protein with a lot of loops and domains sticking out. And what people started to do is called structure-based functional analysis of RNA polymerase, basically chopping off the pieces of RNA polymerase and looking what happens. And what we did, at that time, and it was my first independent project, we chopped off the loop that was not resolved in the crystal structure, which was fairly far away from the active center, but we were just interested what it was doing. And the numbers that we've got from the catalytic activity of RNA polymerase, this mutant RNA polymerase very much resembled what we saw with wild-type RNA polymerase inhibited by streptolidinogen. We indeed went on to show that streptolidinogen acts through the RNA polymerase somehow, through the trigger loop somehow, this flexible domain that we chopped off, and it acts on the catalysis of the phosphodiester bond formation. So it doesn't interfere with the other steps of the nucleotide addition cycle. The question was how it happens. So this is just the space field model of the active center. These are 2 magnesium ions over here, the incoming nucleotide, this is RNA. And the trigger loop, this flexible domain that we chopped off, is this gray blob over there. And this is the streptolidinogen molecule bound to it. So both of them are very, very far away from the catalytic magnesium. So it was unclear how possibly can these entities affect the catalysis by RNA polymerase. But looking at the structure, we suddenly realized that it is very open. And it is very unusual for enzymatic reactions to have the active center that open. You need some intimacy. So a lot of you would expect a lot of contacts of the protein to the substrates. You also would expect the sort of a compact structure which would exclude the free water molecules and ions that are browsing around and that can poison the reaction. And what we hypothesized was that there should be some closure of the active center happening. We suggested that the structure that we observe is sort of open one, and it is catalytically inactive, and it is stabilized by streptolidinogen, and this is the way how streptolidinogen acts. But there should be some sort of closed structure, and this closure somehow orchestrated by trigger loop, where catalysis can be efficient enough. And just 2 years later, both in eukaryotic RNA polymerase and bacterial RNA polymerase, crystal structures revealed the new conformation of the trigger loop, which showed that it can be much closer to the catalytic center of RNA polymerase, bringing some of the amino acids of the trigger loop in the contact distance to the incoming nucleotide. And it is more obvious in the space field model. So this is the open active center that is catalytically very inefficient. And the trigger loop is gray over there. It is an open state. And this state is stabilized by streptolidinogen. And this is the closed conformation. And you can see how much more compact the active center now and how the amino acid content of the active center changes. So we went on to show that histidine here and arginine over there, they participate directly in the catalysis by stabilizing the transition state of the reaction. And you would ask why this complication, why this complex mechanism would evolve instead of just motionless catalysis by the two-metal ion mechanism. The fidelity of transcription is a critical property of it. Copying of genetic information should be fidel. And what we managed to show is that the motionless active center, so the one that, for example, doesn't have trigger loop or the one that is inhibited with streptolidinogen, can discriminate against the incorrect nucleotides very poorly. So it is just 10 to 100-fold discrimination against non-complementary nucleotides and no discrimination against deoxynucleotides. And this sort of discrimination you would expect from just differences in base pairing between the nucleotides. So Watson-Crick base pairing against non-Watson-Crick base pairing, meaning that the motionless active center doesn't add anything to the fidelity of transcription. However, in the case of the mobile active center with the catalytic domain being flexible as trigger loop, the discrimination increases enormously to almost 6 orders of magnitude discrimination against non-complementary nucleotides and up to 10 to 5 discrimination against deoxynucleotides. So why would it happen and how it works? So what we managed to show is that the folding of the closing of the active center, the closing of the trigger loop, is induced by the correct nucleotide binding in the active site. So when the incorrect nucleotide binds in the active site, the non-cognate one or the non-complementary one or the nucleotide that lacks some particular chemical groups like deoxynucleotide, the trigger loop just fails to fold. Failing to fold means no catalysis. No fold, no catalysis. So the incorrect nucleotide blocks the conformational change that is required for catalysis. This is not very new, and it is called the induced fit mechanism of Fidelity, and it is used by a number of enzymes. But now we show that multi-subunit RNA polymerases also use it. And there is just an intermediate conclusion. So the trigger loop is a new catalytic domain which acts along with the catalytic magnesium ions to synthesize the phosphodiester bonds. It speeds up the reaction 10,000 times, and it determines the fidelity of transcription. Since the amino acid content of the active center is so abruptly changed by the folding of the trigger loop, by the closure of the active center, we will refer to the open active site as 0, 1, and it actually resembles a little bit 0 from this point of view. And this active center, when the closure occurs as a trigger loop active center. And there are some interesting things also bound to the flexibility of the trigger loop that we discovered, and I'm going to talk about next. So besides adding nucleotides, besides synthesizing RNA, RNA polymerase can also cleave off mononucleotides and oligonucleotides by hydrolyzing phosphodiester bonds of the RNA. To do so, the RNA polymerase shifts backward, so the active center shifts backward to the upstream phosphodiester bonds, where it performs the catalysis. And the cleavage reaction is catalyzed by the same active center as the addition of nucleotides. We showed that the trigger loop, this new catalytic domain, is also required for this reaction, and it speeds up the hydrolysis by more than 2 orders of magnitude. But what we observed was something unexplainable. This is the only gel, so don't worry, this is the only gel I'm going to show. So second phosphodiester bond was cleaved much faster than the first one. This is the denaturing polyacrylamide gel, where the products of reactions of the hydrolysis reaction is resolved, and this is the 13 nucleotide long RNA, which in time is cleaved by the RNA polymerase active center. But instead of cleaving one nucleotide, it for some reason cleaves two. So the second phosphodiester bond for some reason is much faster, cleaved much faster than the first one. The question is why? So the second phosphodiester bond is cleaved in the so-called backtracked state. So RNA polymerase, as I said, can move backward. The 3' end nucleotide of the RNA disengages from the template of the DNA and is flipped out. And the active center is shifted to the second phosphodiester bond, which is sessile, which will be cleaved. So we started to investigate the reaction, and what we found is that the properties of the reaction depend on the identity of this flipped out 3' nucleotide monophosphate. This is just an example, so just don't worry too much. It's dependence of the rate of the hydrolysis from the pH of the reaction, and you see that they are quite different for each of the possible nucleotides in this position. We went on and we made many different chemical substitutions to this nucleotide, and what we showed is actually that this flipped out 3' end nucleotide of RNA directly participates in the cleavage of the second phosphodiester bond of the transcript. So this becomes a bit funny. So first of all, we know that the catalytic domain of the active center directly participates in the reaction. But the 3' end nucleotide of the RNA that is flipped out also adds up to this active center, so becoming a part of the active site. And it will be more obvious on the structural picture. So again, the open active center, the trigger loop active center that catalyzes the synthesis of RNA, and this is the trigger loop plus 3' end nucleotide of the RNA active center. And the nucleotide is in dark gray here. And you see the marked differences in the chemical groups that make up the active center. So why would this mechanism evolve at all? And if you think about the misincorporation event, which must happen sometime, this nucleotide is erroneous, so it is not complementary to the nucleotide in the template strand. It means that this base pair is very unstable and wants to break. When it breaks, the erroneous nucleotide wants to flip out. When it flips out, RNA polymerase shifts backward, forming a backtrack complex. And as we now know that in backtrack complex, this flipped nucleotide helps RNA polymerase to excise itself from the transcript. So it is sort of self-correcting mechanism of the transcription. Besides misincorporation events, backtracking can happen on some sequences, which I'm not going to go into in details, but the important thing is that this sort of backtrack complexes, they cannot extend it because the 3' end of RNA is away from the active center, meaning that they cause pauses. And causing pauses can be very detrimental to cells because they cause traffic jams of RNA polymerases, queuing of RNA polymerases behind the paused RNA polymerase. And they also can cause the collisions with replication forks. So we think that this mechanism, this transcript-assisted hydrolysis, when the RNA assists RNA polymerase to correct itself, is sort of an ancient mechanism that was there to resolve these backtrack complexes. And indeed, we recently showed that RNA polymerase 2 also uses the transcript-assisted hydrolysis to resolve the backtrack complexes. So assuming that this mechanism dates back before to last common ancestor before the divergence of bacteria and archaean eukaryotes. So this is just another intermediate conclusion that the flexible active center, the trigger loop active center, can be further modified to switch its activity from synthesis to RNA hydrolysis. Okay, so as I mentioned, the backtracking can be very detrimental to living organisms because of causing the collisions and traffic jams. Even though the transcript-assisted cleavage might have been efficient enough to do the job in the last common ancestor, or maybe early after divergence of bacteria and eukaryotes, there is no surprise evolution led to invention of some extra mechanisms to deal with the backtracking complexes. So evolution invented the cleavage factors, which further assist RNA polymerase to cleave away the backtracked RNA. These cleavage factors, GRI-A cleavage factor in bacteria, and well, TF2S is the cleavage factor of the RNA polymerase II in eukaryotes, but all of the archaeal and eukaryotic cleavage factors are homologous. They have nothing to do with each other, so they're evolutionary interconnected, again arguing that they have emerged after the divergence of bacteria and archaeal eukaryotic lineages. Right, so if to add GRI to the cleavage reaction in vitro, the reaction is speeded up more than 2 orders of magnitude. And given our findings about the trigger loop and the 3' end of RNA that I just told you about, we were interested how GRI factor can collaborate with or cooperate with trigger loop and the 3' end of RNA in the active center to speed up the reaction even further. So what we did, we used our favorite enzyme with the deletion of the trigger loop that, again, I purified many years ago. And wild-type enzyme, and compared the cleavage GRI-A-assisted cleavage reactions with these 2 enzymes. And to our great surprise, the rates of the reactions and the properties of the reactions assisted by GRI-A were almost identical for wild-type enzyme and the mutant enzyme, meaning that GRI-A-assisted cleavage did not require trigger loop at all. And this was the first reaction we observed for RNA polymerase that doesn't require trigger loop. So what we hypothesized was that maybe because of the trigger loop is so flexible, it may adopt some sort of open conformation and let the GRI factor into the active center to bring its own catalytic activity. And our hypothesis was supported by, fairly soon, by the crystal structure of RNA polymerase with the homolog of GRI-A. And indeed, the trigger loop in this complex is in the open conformation, catalytically inactive conformation, and GRI literally substitutes for the trigger loop in the active site. It can be more vivid in the space field model. So this is the trigger loop active center that is sufficient in catalysis of phosphodiester bond formation. This is the composite active center of trigger loop and the 3' end of RNA, which is relatively efficient in RNA hydrolysis. And this is the GRI-A active center. So GRI substitutes for the trigger loop in the active site, and you can see the huge differences in the amino acid content in the active site. So this can be, and because both of them, trigger loop and GRI, have absolutely different catalytic activities, you can count it as an exchange of the active centers. And quite uniquely, this exchange of the active centers happens in the same place in space relative to the reactants in the active center. It is very different, for example, from DNA polymerase when the exonuclease reaction to correct the misincorporation events resides somewhere far away from the polymerase active center. And the substrate, so the DNA, has to travel up to 40 angstroms from one active center to another. Here, the RNA sits in the same place while the active centers come to deal with it. We went on to show that actually GRI does not dissociate from RNA polymerase while swapping with the trigger loop in the active site. And you can count it as RNA polymerase carries 2 active centers all the time, which are swapping for each other near the reactants. But importantly, we showed that the exchange of the trigger loop for the GRE factor happens only upon misincorporation or backtracking. So there is a very tight control of the GRE activity. So upon misincorporation or backtracking, the trigger loop is replaced by the GRE, but after the hydrolysis took place, the trigger loop takes its place back and substitutes for GRE in the active center so that the catalysis can, the processive RNA synthesis can continue. And it has a huge biological sense because if you imagine the hugely effective hydrolytic activity of GRE active center being not controlled, all your RNA will be degraded straight away. Okay, and so I would like to summarize all what I spoke about and show you the different active centers now in one slide. So we have the zero empty center, active center, which is insufficient in any sort of catalysis. We have a trigger loop active center, which is very efficient in synthesis of phosphodiester bonds and elongation of RNA. We have a composite active center of trigger loop and 3' end of RNA that is relatively efficient in hydrolysis of RNA, which is very important for resolving backtrack complexes. But because the resolving of backtrack complexes is so humongously important, RNA polymerase acquired an extra catalytic domain, an extra active center, GRE-A active center, and you can see the differences in the amino acid contents there. E. coli has a second, and some other bacteria have a second cleavage factor called GRE-B. It also hydrolyzes RNA, but it does it in more backtrack complexes than GRE-A. So when RNA polymerase shifts backwards by more than 4 or 5 nucleotides, GRE-B will be the one that's to go for. And again, you see that even though they catalyze one and the same reaction, because of the differences in the signals that they are involved in, the amino acid content of the active center is completely different. Small molecules also can modify the active center. So we recently showed that antibiotic Tagetik toxin binds very close, well, you can't see it here, but it binds over there very close to the catalytic magnesium ions, and it modifies the trigger loop active center. You probably can see some marked differences, like this phenylalanine is flipped out a little bit, and some groups are shifted. However, quite amazingly, it doesn't change the catalytic properties of the trigger loop active center. Instead, it delays post-catalytic translocation of RNA polymerase. So when RNA polymerase wants to move forward to vacate the active center for the next nucleotide, Tagetik toxin delays this reaction by altering the active center. Okay, so the more the merrier. So we have two factors, GFH1 and DKSA, which are also homologs of GRE factor. And this is, I just fitted them in the structure, and you see that also they also provide very much their own active centers to RNA polymerase. We don't know what GFH does in vivo. In vitro, it slows down transcription a little bit, and it inhibits also the GRE catalyzed cleavage. But it doesn't bring any other activity on its own. DKSA is a global regulator in many bacteria, global transcription regulator in many bacteria. But how it works, the molecular mechanism, how it works, we don't know. And also, we do not see any activity that DKSA brings to the active center of RNA polymerase. However, by analogy with GRE factor, maybe we are just lacking knowledge of the signals that really force DKSA or GFH to substitute for the trigger loop in the active center and bring their own activities. So there are other three homologs of GRE factor called RNK, TRA, and YACL. We have no idea what they do. So in vitro, they are very silent. In vivo, you can delete them easily. But yet again, we just don't know the signals that force them to come to the active center and substitute for the trigger loop. The Alamon PPGPP crystallographically was shown to be able to bind in the active center, which assumes that it may also modify the active center to change its properties. The antibiotic microcin J25 is a peptide that binds also in the vicinity of the active center, and it modifies the trigger loop active center so that RNA polymerase becomes more responsible to pausing. We don't know the molecular mechanism for that, but yet another active center. So as I said, the archaea and eukaryotes have their own cleavage factors, which are unrelated to GRI factor, TFS in archaea, and these three are in three eukaryotic polymerases. They are all homologous to each other. They were shown also to act by substituting for the trigger loop. So yeah, and the alpha-amanitin is the inhibitor that the antibiotic that inhibits the RNA polymerase II. It also binds at the top of the over there at the top of the active center and sequesters the trigger loop active center in a conformation that is incompatible with catalysis and translocation. So why I'm showing that is to show you that the substitution of the active centers is something belonging to all domains of life. So it is something very ancient and very profound. And this is the model that, the working model, that RNA polymerase has several active centers that substitute for the trigger loop active center in response to some signals. Some of the active centers may be bound to RNA polymerase permanently. So assuming that RNA polymerase carries several active centers with it, some of them can come from the solution to substitute for the trigger loop or to each other upon the changes of the signal. Right, I wouldn't bore you with the conclusions. It's just the major points. The major conclusion is that the active center of RNA polymerase is not that boring as we thought. The trigger loop is the new catalytic domain that determines, that is required for catalysis and determines the fidelity of transcription. And the flexibility of the trigger loop allows factors to modify it or to substitute for it and thus change the properties of the active center of RNA polymerase. And just in conclusion, I would like to mention the people who were involved in the work. Konstantin Severinov in Rutgers University was my PI for the part of my PhD studentship and a postdoc. Yulia Yuzenkova was my postdoc here in England, but now she has her own lab as she became the Royal Society Research Fellow. Sasha Bachkarova and Soren Nielsen are postdocs in my lab, and Mohamed Raganyan was my PhD student in my lab. And these are the funding bodies that were at some point were involved in the project. And I thank you for your attention.
I think one of the great things about the Fleming Prize lecture is you hear some great science, and you know there are many, many more years of great science to come, and we look forward to hearing about that in due course, Nikolay. So it's a great pleasure to present you with the 2014 Fleming Prize Award.